So as you all know, we'll be going over machine learning for beginners. Everybody can see my screen fine, I assume, otherwise someone would stop me. But yeah, feel free to, unmute anytime or just send a chat. But yeah, let's get into it. So the difference between machine learning and traditional programming. In traditional programming, right, we have lots of data, we write rules, also known as our program, and feed that into a computer and generate outputs. Whereas in machine learning, we have all our data and we have our known outputs and we feed that into the computer and get the rules or a program. And this is our output model. So yeah, so we want to gather information about how we go from data to output. And this can be applied for a few functions, but in general, we describe them as descriptive, predictive, and prescriptive. So descriptive, getting information about what the data explains, predictive, using the data to predict what will happen next, or prescriptive, using the data to generate suggestions. So going over the main types of machine learning that we use to do these tasks, I'll be talking about unsupervised, supervised, and reinforcement learning. So one by one, first we'll talk about supervised learning using this example of cats and dogs. So essentially in supervised learning, this is for when we have labeled data. In this example, we have the label of cats. These are all cats. And we feed that into the machine or the model, and it learns that these images are cats. And once we have that trained model, we can feed in, any image, in this case, a bunch of cats and dogs, and the machine will let us know these are cats and these are not cats. So this is an example of classification where we sort things into these like qualitative categories. Supervised learning can also be done using quantitative labels, and in that case, that task is usually regression. This can be used for like, predicting the weather, or like the housing market, stuff like that. So next we have unsupervised learning with the same cats and dogs. So in this case, unsupervised learning is when you don't have labels. So here, yeah, none of these cats and dogs are labeled. We put them into our machine learning model, and the machine sorts out the images into groups. And they don't give us the names of those groups, they just cluster them. But once you see the clusters, we can see that they sorted them into cats and dogs for us. So that's one of the tasks that can be used, that you can use unsupervised learning for, is clustering. And then another example is anomaly detection. So if one subset of your data is like abnormal compared to the rest, it can also do that. And then lastly, reinforcement learning. It's kind of like dog training. So yeah, here's this dog. In which case, let's imagine, the dog is the model or the machine. It can go between two states, sitting and walking. And based on the environment, let's say we want it to walk. So when it's walking, we give it a reward so that next time it knows in this environment you should walk. And that's kind of how the process of reinforcement learning works, is the machine does an action in a certain environment, and that updates the sensory inputs. And depending on, whether that's good or bad, we assign it a reward weight so that it can perform certain actions given the environment. And this can be used for a lot of things, but I think, the most fun version is like we can train computers to play video games, beat people in chess, stuff like that. But yeah, this is reinforcement learning. So next, you know your task, your data set, which algorithm or like model do you choose? This is kind of, I know, an overwhelming image, but if we just go through it slowly, this is a cheat sheet from the library of scikit-learn, and you start the starting point, and based off of like how big your data set is and what kind of task you do, you can follow the flowchart to find these boxes in green, which are the machine learning algorithms. And you can usually start there and see like how the model does and the accuracy and stuff, and then decide like where else you should try. But in general, when choosing an algorithm, you're really thinking about, what is my data set composed of? Is it text? Is it images? Is it audio? And like, how big is my data set? You might want to know, like, you should probably have at least 50 samples no matter what. That's really also kind of small. Just something to keep in mind if you're like an experimentalist and you want to generate your own data, like it would benefit to, have some kind of high throughput process to at least get 50 or 100 samples to begin with before you start implementing machine learning. But let's say we have a small data set and we want to make it bigger. We can do data augmentation, where that's essentially we just transform the data we have. Here's an example with images. This is the original image, and we just augment it by rotating it a bunch of times. And each one of these images can then serve as one sample. So we essentially, increased our sample size by 4. And this is useful to prevent overfitting, but also, in this case where we have too small of a data set. And you can really augment any type of data. I guess these 3 types, here's some like good examples of how you would augment them. So audio, you can like add noise, change shift and speed. Text, you can just like move the words around. And then images, you can do all kinds of geometric and color transformations, and also like add noise and stuff. So we'll go over this a bit more later. But in general, once you have, your data set augmented or not, and you want to put it into the machine learning pipeline, you first train your model, and then you have to validate it and just see how it does. So how do you do that? So to do validation, there's 2 main ways you can approach it. So holdout validation is where you just split the data set into a training, testing, and validation set. So you just subset it and you train on your training set, and then get your accuracy and validation metrics from your testing and validation set. And this can be efficient for if you have a really large data set, or if you're just like starting out and want to test a bunch of models and see like which one you should go with. This is a pretty quick and easy way to get a general gauge of accuracy. But the more robust way is to do cross-validation. And that's where we split the data set into K number of groups and then repeatedly test and train using each unique group. So here in this image, this is 5-fold cross-validation. So you can see we split the data set into 5 groups and train it 5 times. And each time we change the test group subset. And then the most extreme version of this is leave one out validation, cross-validation, where K, the number of groups we split it up into, is equal to N, the total number of samples in our data set. And so this is, I guess, like, top tier validation. But as you can imagine, if you have hundreds of thousands or even like tens of thousands samples in your data set, this would take a long time. So that's where, holdout validation would come in handy. So that's how we get our validation and accuracy. So then we get all these metrics, right? Like which one's the most accurate, how do we, make our accuracy better? We got to find our best parameters. So before we get into how to optimize our parameters, a little, I guess, syntax thing. In machine learning, there's hyperparameters and parameters. So our parameters are the like variables in our model that the machine will adjust itself to when it's training on your data. Whereas the hyperparameters are the variables that govern the way the model trains, and these are the ones that we can manually adjust. So you can manually, just go in there and test them, but a more efficient way is to do a search, specifically either like a grid search or random search. A grid search is where you define a range for 2 separate parameters and essentially just test all of these combinations and see which one is the best. In this figure, the white area is what we would consider the optimal parameter range. And as you can see, sometimes if you do a grid search, it'll miss it. So that's why sometimes we want to start off with a random search where you just randomize the 2 hyperparameters and see if you get one kind of close to the optimal range. And then you can do a subsequent grid search like in this area. But yeah, these hyperparameter searching functions are typically built into libraries, so it's not too complicated to implement and much faster than just like manually guessing, But yeah, that's how we get our parameters optimized. And yeah, so now you know all of this. How do you actually do it? You can choose really any programming language that you are comfortable with. There's machine learning capabilities in almost all of them. And there's also, you have to choose like what work environment you want to use. So these integrated development environments are just like easy like user portals for coding. And these are some like popular ones if you aren't already have experience in coding that you might want to check out to use. And then platforms. So to run the training, usually, most people don't have really strong CPUs or GPUs to run training on. So we usually run them on these servers, but usually that's built into whatever development environment you're using. So don't worry too much about deciding on these. Just choose, a language you're comfortable with and a setup and go from there. And then here's also some beginner-friendly resources that you can look at. So here's some beginner-friendly libraries, scikit-learn and Keras. These are both in Python, and these are all linked. So if you're in the read-only PowerPoint, you can click on them. And then there's also this caret package in R, which is good for image classification and regression. Also, R has a lot of like built-in things into their base language that is good for machine learning. And then here are some classic data sets that are like really popular. If you, take an online course in machine learning or any course, you'll probably end up like using one of these, but CFAR 10 is right here. It's a bunch of images with these 10 different categories. And then ImageNet also has labeled images like this, but there's also like different levels of labels. So there's image level where it says, there are or not this object in the image. And then there's also like object level where they label objects in the image and like give you the position if that's something you're interested in. And then MNIST is this data set here where it's a bunch of handwritten numbers, and usually people try to classify the numbers based off that. And then also I want to give a plug for Kaggle. This is just a website / community that has a lot of resources. There's 50K plus public data sets on the website. They have courses that I think you can get certifications for. And they also have like lots of public notebooks with examples and a really active forum and community if you wanted to get involved or get help. They also like throw competitions and stuff that you can enter. But yeah, that's all the resources. And then now we can move to the Colab tutorial where we're going to show how to build a binary classifier. So that means just two groups where we identify histology images with sickle cell anemia or healthy. And we go over pre-processing the images. So how do you convert them to the right format and resize them? Then we label, and then we show you how to test various scikit-learn models and tune them. And lastly, a little bit on data augmentation. So if y'all want to click the link in your screen, I will shift over. Tahseen will share his screen now.
Great, thanks. Can you see my screen?
Yeah, we can see.
Great. Okay, great. So yeah, in this section, we wanted to take you through a typical coding pipeline of training a machine learning model. So as Kayla mentioned before, for typical validation of a model, let's say you're trying to publish a paper about some new result, you're usually going to basically assert the validity of your model using something like MNIST or CFAR10. But we thought it would be interesting to actually curate and build up a custom data set, which happens a lot in real industry applications. So we're going to essentially build a binary classifier, as Kayla mentioned, which will identify whether or not a histology of blood cells presents for sickle cell anemia or is healthy, okay? And just go through the process of actually building up that data set from scratch. There's a lot of machine learning. The bulk of machine learning, I would say, when you're working with custom data sets, is actually pre-processing that data set so it's ready to plug into a model. Plugging it into the model, once you get to that part, actually isn't so bad, especially if you're using a package like scikit-learn, which I'm using today. Okay, so I think the one we're sharing is read-only. There should be a copy in there that you can download as well. You're free to execute these cells as you like. Some of them will take a while to compile. So I'm not going to execute anything here today, but rather just walk you through the pipeline and what code is going into each of these blocks, okay? So we're working through Colab today, which is an excellent resource for basically collaborative coding of any kind. What's great about these notebooks is that they come preloaded with a lot of essential machine learning packages, but you can also install a lot of custom packages as well. So in this first block, I'm going to install some packages that I will need later on, but then the code block below it is just me importing all these things that I'm going to need later on. And then one important thing here, and this is specific to Colab, if you want to use folders within your Google Drive, as I will need to do, you just need to mount your drive, and that amounts to nothing more than just giving this notebook permission to see your drive. And even if you hand this notebook to someone else, it's not like they will be able to see the items in your drive. It will just attempt to mount theirs. But I'm going to need this to actually get all the images I've downloaded. Okay. So yeah, so the first thing I'm going to need to do is actually download the images I need for my two classes. So my two classes are sickle cell histologies and healthy red blood cell histologies. Okay. So how do I do this? I simply go in Google Images and then I download a bunch of images, sickle cell histologies and healthy cell histologies to my drive. And the better structured or the better separated your images are through clever folder structure, the easier it will be to process, pre-process these images. So when I was first downloading my class consisting of healthy blood cell histologies. I put them all in a folder named healthy. And then likewise for sickle cell images, I put them all in a folder named sickle. So it would be easy to loop through these folders and naturally separate my classes as I'm processing them. Since we're only using scikit-learn, we don't need to use fancier data loading as you might need to do with libraries such as PyTorch or TensorFlow. But if you are using those and you want to use the so-called data loaders in there, which prep your data for training, your folder structure is actually going to be a little bit more complicated than this. Presumably because it assumes you're going to be using a ton of images, you're also going to need folders which basically separate your train images from the validate images from the test images. Okay. So yeah, so basically I went through Google and I clicked through a bunch of these histologies and I downloaded them. All right. And then once they're in my folder, since I've mounted my drive, I can actually loop through a directory and add them each to their own array. Okay, so I'm going to create two arrays consisting of healthy images and sickle images, and I'm going to load them in. Okay, here I'm using a package called Pillow, which is really nice for processing images and converting them between NumPy data arrays, which you could think of as matrices or tensors. So I'm going to use Pillow to read through all the files, which are JPEGs, open them up, and add them to an array as a NumPy data array, okay? All right, so you might be wondering, okay, so when I look at an image, it looks two-dimensional, right? It's height and width, but if I were to convert it to an array, say pixel values, what is the actual shape of that? Well, when you do it, you're actually going to notice that the shape here, sorry, it didn't spawn here, but it's actually going to be height, width, and color. So it's going to look like a three-dimensional array. And why is that? Because when Pillow converts an image over to NumPy, it's going to assume the image is on an RGB scale. So if you think of an image, even a JPEG, what's actually going, so you have basically the height and width of pixels, and you essentially have three slices of it that are in some sense overlaid over each other. The first is the image in red scale, second is the image in green scale, and the last slice of that is in blue scale, okay? So even though you might have a height and width of, let's say, 128 by 128, there's a lot more going on in the image because there are going to be three slices of 128 by 128 corresponding to each individual color scale. That makes sense, okay? So then the actual number of parameters that you have going into an image is essentially height times width times color. So it's a little bit bigger than you would expect it to be. It's a lot more than just height and width. Okay, so at this point, I've basically converted all my images to NumPy arrays and then gone into my array. But for training, other than language training, which we don't cover here, you pretty much need all your data points to be standardized. Okay, so if it's a feature vector, you need each of your data points to have the same number of features, or at least be the same length. And the same for images, I'm going to need them to be all the same size. So the first thing I'm going to do is I arbitrarily decided that I need every image to be 32 by 32 in height and width. We'll still keep that color depth to it, but I'm going to resize the height width, okay? And that's accomplished using a simple resize function that comes as part of Pillow. So this is the image blown up. So obviously, if I shrink an image and I try to zoom it out to the original scale, it's going to look a little bit blurry. But if you were to take these images and look at them as a 32 by 32 image, it's going to look pretty clear, just tinier, right? And a lot of resizing an image actually makes it easier for your machine learning model to process. For instance, for the MNIST data set that Kayla was talking about, those don't even have color depth. Those are just black and white. Okay, so you sort of lose that third color dimension. And since they're 28 by 28, they're pretty small, so they're easy to process. And there's about 60,000 of them, right? So time is a factor we also need to consider. So keeping images smaller will greatly improve training time. All right. So now we have all our images resized. It's a little blurry, but you can kind of tell where some of the sickle cells are. But our model will definitely be able to tell at least. All right. So now that I have my images, they've been resized. Basically, what I'm going to do is I'm going to smash them together, and that's going to be one total data array. Okay, so it contains all the healthy cells and it contains all the sickle cells. Okay, and then I need to develop another array, okay, which is labeled here as Y, which contains all the label. Okay, so in my data array, if I were to basically look at my data array and look at my label array, if basically the nth data point in my data array, you would be able to identify the label of that by looking at the nth data point in my label array. Okay, so there's a correspondence between these label arrays and the data array itself. So basically, in the label array, it's pointing a label to the actual image at the same position in the data array. That makes sense. Okay, because the machine learning models, the supervised model, will need both the labels and the data. Okay, so from here, I'm going to create a general machine learning function that will allow me to plug and play different models. Okay, and we're not going to look at this. We're not going to go too deeply into the code, but as a high level overview, first thing I actually need to do is for these scikit-learn models that we will be using first up, they typically prefer vectors of features. So if you think about a 32 by 32 by 3 image, I can basically take all those color slices, unwrap them as a vector, and just stack them as each other. Okay, and 32 times 32 times 3 is a little over 3,000. So really, we have 3,000 features here. So I'm going to unravel that image and basically present it as a vector, okay, for each of those. And that's what we're going to feed in to the machine learning model. Okay, and I've also put in an ability to define the extent of folding through your cross-validation. For most of the stuff I will be doing today, other than the actual neural network at the end, I will be doing the leave one out cross-validation, meaning that your training set is all but one image, and then your task set is one image. So this is, in some sense, the most aggressive form of validation, because with larger folds in your holdout, especially if the data set gets really large, there's no way that you can essentially do leave one out validate cross validation, because you'd be trained. For instance, if you had 50,000 images, you would basically have 49,999 rounds of training using basically the full extent of that data set except for one. That's going to take too long. But for us, since our data set is small, as you can see here, we have 86 healthy images and 68 sickle cell images. It's not too bad. Leave one out is going to finish in a relatively timely manner. Okay. And then for each of these models that I fit in, so basically you define the model, you fit it to essentially your training data set and your training label set, and then we're actually going to make a prediction. And then for each cross-validation, we're going to record whether or not the model is successful at identifying the status of the histology left out in test, and we're going to measure the total accuracy over the data set. So at the very end here, I'm going to print out the total accuracy of our model and also present a confusion matrix, which basically tells us, well, how good is it at predicting each individual class? If you just look at raw accuracy, that doesn't really tell you the full story, because you are actually interested in identifying if the model is struggling on a particular class. Okay, so for us, I want to see if our models are better at identifying healthy images or sickle cell images. Okay, so the first very simple model I'm going to run it through is K nearest to neighbors. So essentially, if you were to look at all the images as a vector, they all have essentially a real value between 0 and 255. And you can take a distance between these vectors. You can think of that as L2 Euclidean distance, and then actually measure similarity between two image vectors in terms of their L2 distance. Okay. And then what I'm telling my nearest neighbors to do is to look at, so for a given image, I'm going to look at its 7 closest neighbors in L2 distance. And whatever the majority class is of those 7 neighbors, I'm going to say that image also falls into that class. So basically, we're using labels to cluster out each of our data points, okay? And then if you do this, if you basically try to classify an individual image using the majority class of 7 of its closest L2 neighbors, it's not that accurate, right? As you can see here, it's about 58% accurate, which is, if we were to just randomly guess, the majority class there makes up 86 out of the 154 total images. So this thing is about 3% better than just randomly guessing, right? Which is not that great. If you look at the confusion matrix, it's really, really good. So the first row basically corresponds to healthy images, and the second row corresponds to sickle cell images. So according to the first row, nearest neighbors is pretty good at telling whether or not an image is healthy, but very, very bad at telling whether or not an image has sickle cells. You can see here, 4 of the healthy images get classified as sickle cell, but 60 of the sickle cell images get classified as healthy. So for whatever reason, this is probably the first machine learning model you try out anyways, but it's not great at basically separating these 2 classes. Can you say, oh, thanks for the question. Can you say again why you went with 7 for KNN? Because I'm hiding a lot of stuff here and I tried out different neighbor sizes. I went through 2 to 10, which is the typical sliding scale that you would usually go for. And it didn't get much better than this. So this is about as good as you get. Typically, you're going to see some sort of slight increase as you increase the neighbors, and it's going to dip off very quickly. Okay, thanks for your question. All right. So now we're going to feed it through a random forest classifier. I'm not going to go through too much of the details there, but gradient boosted machines and random forest classifiers are essentially very intricate decision trees. Okay, and the way those trees are built up are what make up the difference between random forest classifiers and gradient boosted machines. But long story short, the rules they use are more than, say, just L2 distance that you would see in K nearest neighbors. Basically, each of the decision trees, if you were to look at the branches, it's going to make a decision based on varying qualities of the individual features themselves. Okay, so with a decision tree, you can control things like depth, even the amount of rules that are at each depth level. But for us, I am going to use basically all the default settings for random forest classifier. And for decision tree models like random forest and gradient boosted machine, what you can't see here is that essentially both of these types of models are going to take the vote of many different trees. Okay, so for random forest classifier, it's actually, we're essentially taking the majority vote of 100 different mini classifiers that we sometimes refer to as weak learners. And it's not easy to print out the rules that each of those voters are going by to make their final vote for whether or not a class is healthy or sick. But by increasing the number of voters that are allowed there, and the default here is 100, if you just pipe in random forest classifier, by increasing the number of estimators, that can sometimes improve your accuracy. But we're going to go with just the default of 100 weak learners. Okay, so once I feed this data set, once again, leave one out cross-validation, through a random forest classifier, now we have a model that's actually pretty decent. It's about 78% accurate, which is a lot better than the 55% accurate that we would have achieved if we were just guessing randomly, right? And then if I take a look at the confusion matrix here, you can see that this model is basically, for each of the classes, it's able to predict each of those classes by a majority that's 50% or greater. What I mean by that is essentially 73 out of the 86 total healthy images are classified correctly. And then 47 out of the 68 total sickle cell images are classified correctly, okay? So this is, there isn't as much mix-up as we saw with K nearest neighbors. So this thing, we already have a classifier that's pretty good at identifying whether or not a histology has sickle cell anemia or is healthy. Okay, let's see if we can do even better. So the next thing I'm going to use is referred to as a state vector machine. This is good for, well, there's several situations you want to use in SVM, and there are actually different shapes of the so-called kernel that you can go through. Here I'm going with a linear shape, but there are other shapes such as polynomial, sigmoid. There's another thing called radial basis function. So if you imagine, so the SVM is great for unsupervised learning, but if you can also determine how well it is at clustering data if you have labels. So essentially, this is great for the situation where you have more features than data points, okay, which is exactly the situation we're in right now. We have over 3,000 features, but only about 154 data points. And then you would change the kernel if basically the way the data is represented in high dimensional space needs to be separated essentially a certain way. So typically you're not obviously going to know what the essential separation of these classes look like in high dimensional space, which is why normally you would feed in a lot of different kernel shapes until you got decent accuracy. So for us, I didn't show the process of going through different kernels, but linear seemed to work the best. Okay, it was about rounding up 79% accurate, so a little bit better than the random forest classifier. And once again, we have great, decent classification for sickle cell, but great classification for healthy. Okay, so at this point, even though the SVM did the best, the model I'm going to fine tune is the random forest classifier. Only because with the SVMs, there isn't too much fine tuning you can do with it beyond just deciding on the kernel shape. There are things you can do that I would say are a little bit more subtle, like if your kernel was of a polynomial variety, you can control the degree of it. But the random forest classifier in our case is going to have a few central features that are easy to fine tune through. All right. For fine tuning, I'm going to use a package called Optuna to do this automatically. All right. You can do this. You can almost hard code this by using some classes in scikit-learn, but using a package to automate this is a lot easier because then you can just sit back and relax and then come back to it maybe in an hour or two, and it'll tell you the best combination of hyperparameters. So with Optuna, there isn't too much that goes into it other than you have to define a function that basically has two components. One, it takes in a trial object, which is specific to Optuna. And the trial object has basically what it does is you first determine what hyperparameters you want to tune. So for me, I'm only tuning 2 hyperparameters. This is just naming it. But as you can see here for n estimation max depth, these are 2 essential hyperparameters for random forest classifier. So question here, my data, if my data low n, high, okay, so has highly correlated features, does that impact the choice of ML method? That's a great question. So, okay, so I'm going to go into, so if you had a low n, so you mean as in a low number of data points, then let's see here. So yeah, you would probably go, if there's strong correlation between your features, then there's going to be essentially more structure the way your data point presents itself in high dimensional space. So I would go with like a state vector machine, or something like that. And high M, I'm assuming high means high number of features. Then that also makes it a great choice for a state vector machine. So that's a good question. Now, if you had high number of n, so if you had a large number of data samples, then it's going to really depend on what type of task you're running. I would say if you had a high number of data points, a high number of features, and say you were trying to do image classification, then you're going to go through something that's called a convolutional neural network. I go through a very basic neural network here, which is known as a multi-layer perceptron. But for an image in which your pixels are correlated with each other, then I would go with something that's referred to as a convolutional neural network. And hopefully we'll get to talk about that towards the end. But yes, great question. Okay. So going back to the fine tuning, right. So you just create essentially a function that returns an accuracy and takes in a trial object. So this trial object basically suggests for the random forest classifier, the number of weak learners or estimators to use. So for here, I say, I'm telling it to pick some number essentially randomly between 10 and 150. And for the depth, each voter is essentially a decision tree. I'm telling it how deep that tree should go. So for here, it's between 2 and 10. Okay, so for every single trial, our objective function, when we pipe it in Optuna, is going to essentially randomly pick a number of estimators and a magnitude to depth every single round. And basically, as the accuracy stays high, it's not going to pick a hyperparameter too randomly. If the accuracy seems to be high for a model, it's going to try to stay as close as to those current hyperparameter selections as close as possible. But essentially, we can think about this as Optuna randomly selecting hyperparameter magnitudes every single trial. So that's it. So you just tell it what classifier to use. You give it essentially a range of assignment for all the hyperparameters you care about. And then from there, you just create the study, and the direction says here maximize, which means that I want you to go ahead and find a model that has the highest accuracy. There are reasons why you want to go into minimize for reasons that I'm not going to get into, but things like adversarial learning, you might want to move in a minimizing direction. But basically all this is saying is like, Optuna, here's my hyperparameter ranges. Now find me the model that performs the best over 100 different combinations of hyperparameters, right? And that 100 different combinations is specified here by n trials. I could go more than that. I could go as high as I like, but each of these trials takes a long time depending on the complexity of your data set and the complexity of your model. So 100 is usually it's usually a good starting spot. Okay. So from here, as you can see here, this is output that I've already compiled. But as you can see here, what Optuna does as it goes through this study, it's going to tell you trial n has finished with, and it's going to tell you the accuracy. So this is why your objective function turns into accuracy, because it just pipes it straight into this output. And what's great about this, each of these trial runs will tell you what combination of hyperparameters it used. So for this first trial, I tried to use 45 estimators and a max depth of 6. And then at the end of each round, it will actually tell you, okay, well, here's the output of this current trial. Here is thus far the best output we've achieved and on what trial it occurred on. So at the very end, you can go back to the best performing trial, dig out those hyperparameter settings, and then use those for your model. Okay. So for here, I ran 100 trials. So actually a little bit less than 100. I think something timed out here. Either my browser, I think my display went to sleep here. So it didn't quite finish. It basically left off 4 trials, but basically 97 trials were finished. And the best trial occurred pretty late, around trial number 82. And with 107 estimators and a max depth of 4, it was able to achieve, rounding up, about 81% accuracy, which is good because now we found that by fine-tuning a random forest classifier, we can develop a model which is 81% accurate on our custom curated data set. Okay. And then from there, I'm not going to do this here, but from there, I would try to take those hyperparameter settings and then basically run the model over and over again and see how often it achieves 81%, because there is some certain extent of randomness. Even if you were to put in these exact hyperparameter settings, the actual output of the accuracy would still be different in real time, just because there's a level of stochasticity or randomness that goes into these voting classifier models. But at least this tells us that this model is capable of achieving greater than 80% accuracy, which is great. Okay. All right. So for now, so that takes care of the simple machine learning models. Okay. For this next phase, I am going to now try to use our data set for actual deep net classification. Okay. And I'm going to spoil it for you here at the very end. We're going to get an accuracy that's better than random, but we're not going to fine tune the neural network a ton, just because I wanted to use the scikit-learn package, and using things like convolutional neural nets, which we would need for this data set, is preferable for getting high accuracy over tasks like that. But I at least wanted to show you what would be the workflow if I did want to use a small data set for deep net classification. Okay. So the first thing I'm going to need to do is, for small data sets, a little bit of imbalance, which is, different class sizes is okay. But for a neural network, classification, we're essentially going to use a data set. We're going to augment our small data set into the thousands, okay? And from there, that imbalance is going to greatly show in our holdout, and we don't want that to happen. So long story short, I'm going to trim both my classes to be the same, okay? So in our original small data set, there were 68 sickle cell images and 84 healthy images. So I'm going to trim off randomly, essentially randomly, just the last 18 images out of healthy. So basically there's 68 healthy images and 68 sickle cell images, okay? And then I'm going to define a function here that's going to come in later, but basically this is going to partition my image into quarters. We're not going to do that yet. So the idea is I have a really small data set and I want to augment it. So basically I turn it into a data set that contains thousands of data points, okay? So the first things first is I'm going to use a library that Facebook released, I want to say about two years ago, called Augly. Okay, and Augly is great for performing simple image augmentations on image files, such as JPEGs, pillow images. It's pretty format agnostic, but Augly makes it easy to manipulate your images. Right. So I'm going to import this library here. And the first thing I'm going to do, the first new set of data points I'm going to create are brightened versions of my original set of data points. Okay. So for here, I'm going to run through all the healthy images and all the sickle cell images, which I've separated just because it'll make it easier to attach those. Are you sharing the code? Yes, this notebook will be available after this. And I think we'll go ahead. I think we shared a public link, but we'll go ahead and send that again, just so you can use this notebook on your own later. Okay. But yes, this notebook will be available. Okay, so for each of the healthy images and sickle cell images, the first augmentation I'm going to do is brighten them. Okay, so I'm going to create a new set of data points, which are essentially brightened versions of the original images. Okay, so this is what it looks like. So if we were to scroll back, let's see, so this is what it looked like originally. It indeed looks brighter. Okay. So these are new data points. So now I have an array which contains regular images and bright images. Okay. The next set of augmentations I'm going to feed it through are just flippings. Okay. I'm going to flip all the images horizontally and flip all of the images vertically. And you might ask yourself, why would we want to do this? Well, if you're a machine learning model and it's trying to study histology, even if you know the general shape of a sickle cell, the way the model is going to see it might be angle dependent. So if I gave you a sickle cell image that was basically upright, a model might classify it differently if basically the sickle cell image was rotated. So doing things like rotations as new data points is really helpful for the learning process. Okay. So basically, it's quite easy. There's just this hflip function with Augly that basically flips all your images. And sorry, this is an older version, but this should have been just a vflip. And vflip will just flip them all vertically. Okay. And then for brightening, sorry, I didn't go over the sub function there, but it's pretty easy. It's just it's a brightness sub method. And then you can control how bright the image needs to be using this factor parameter. Okay, the next set of augmentation I'm going to do is make all our images grainy. So essentially make it a little bit foggier. I refer to it as salt and pepper noise, and this you can achieve using a shuffle pixels augmentation. Once again, there's a factor associated with it. So basically, this will control how grainy the image becomes. So if you look down below the original image, it looks a lot less. The clarity is essentially reduced because I put a lot of grainy noise on top of it. Okay. The next type of augmentation I'm going to do is a blur. So the default method it's going to use is what's referred to as a Gaussian blur, which has a very particular filter on top of it. But as you can see here, the radius parameter inside this blur method controls essentially how blurry the image is going to be. So I played with basically sliding scale parameters until I got something that was moderately blurry, but not too blurry, because at the end of the day, I don't want to destroy the sickle cell shapes that are inside the image. But yeah, this is reasonably blurry. So once again, so basically through this process, I'm forming more and more data points. So this, adds in blurry data points. Then I'm going to skew it. So basically this takes your image and then basically rotates it, or basically skews it at a random angle. So you can see here, this was the random skew angle that occurs. So with Augly, all you do is just feed in, you feed it into the skew sub method. All right, there's no extra parameter there. And then it'll basically skew it at a random angle. So I'm going to add those image augmentations into it. For contrast, okay, so for this next augmentation, I'm going to basically manipulate the contrast inside the image, also controlled by a factor pretty similar to brightness. Now those in here so I can see here. This is increase the contrast in my original image. So yeah, so these are, I would say, most of the common image augmentations you can do with Augly. If you were working with textual data, or even audio data, there's a lot more you can do. There are a lot of interesting augmentations you can perform on data sets of that kind. But for images, this runs through the standard gamut of augmentations that you might use on a set of images. And then, so we're gonna ask yourself, okay, so how much have we expanded our data set? So as you can see here, we've actually gone from 68 sickle cell images to 6,500. So we have greatly augmented the original size of our data set. Okay. And the last thing I'm going to do is every single data point I'm going to quarter, because then that's going to give us basically 4 times the number of data points in each class. So basically, if you look at an image, what I'm going to do is equally cut it into quarters. So basically, we're going to, as data points, we're going to feed in patches. Quartered patches of each image, okay? Because each of those patches should still contain a sickle cell, so it looks like an entirely new data point to the model. So I'm going to create the labels as usual. I'm going to put so basically for each class, we have about 24,000 images by the end of it. I'm going to push them all together. I'm going to assign the labels to each of them. And then we are going to feed it through a multi-layer perceptron, okay? Perceptron. So if you remember, so I think Kayla showed us an image of what a typical deep net looks like. Essentially, by default, it's going to have 100 hidden layers, okay? And there are going to be things like nonlinear activations between them. So with sklearn, the only neural network you can essentially build up is in multi-layer perceptron. You can't build fancy models like convolutional neural networks. That you would need to use PyTorch or TensorFlow, which I won't be showing here. It's a little bit more complicated, but you can at least implement a multi-layer perceptron, and by default, it is 100 layers. But for here, you can actually manipulate the number of hidden layers. So I made this 200. You can tell it, so max iteration here tells you essentially, so we use this thing called stochastic gradient descent to alter the weights of each neuron, and that will greatly affect the accuracy of the model as well. We're not going to go into the math behind that, but what I'm trying to say is with sklearn, even for a multi-layer perceptron, you have a good, you can customize the MLP in a variety of different ways. You can increase the depth of it. You can determine the number of stochastic gradient descent steps. You can control the number, the types of nonlinear activations that go between the layers. There are lots of things you can do with just a multi-layer perceptron. And then for a lot of tasks, multi-layer perceptron might be all you need. So when I feed it through that and I train it, I think it took close to 10 minutes. This is going to take a lot longer for more images and increased task complexity and increased number of hidden layer parameters, but this one took about 10 minutes. And the score without any further fine tuning was 61%. Okay, so better than random, but not quite there yet. So the things I would be tuning there would basically be, I do a variety of things. I would increase the hidden layer depth. I would vary up the type of nonlinear activation that's used between layers. I would vary up the number of iterations. I might even play with the image size, the input size. And then one important thing to note here is that since we had about 50,000 images, there's no way we could have done leave-one-out cross-validation. So basically 75% of those data sets, of the data set was used for training and 25% of it was used for train. So I might even vary up the split and see how that affects it, just to determine whether or not there's some sort of correlation that's happening with my holdout that I don't want to be occurring. So I'd vary up the split, I'd vary up the hidden layer size, type of nonlinearity, number of iterations, et cetera, things like this. And the score might get higher. But ultimately, I think if you did that all, you probably could get up to around 70%. But what I would actually use if I were trying to augment this data set and seriously train it, I would use something that's known as a convolutional neural network, which is great for image processing. Fortunately, you can't use it in scikit-learn. It's a little bit more advanced and you have to build it up using PyTorch or TensorFlow. But at least we at least want to communicate that using scikit-learn, we can at least implement simple versions of deep nets. And who knows, maybe with fine tuning, you get it up to 80%. I'm not really sure, but that's why we're giving out this notebook so you can play with it yourself. But yeah, so that is a typical workflow of how you would go from a custom data set to training a machine learning model to identify classes within that data set. Okay. I think we finished just a little bit before time is up. We're happy to take any questions now. And as we said before, this notebook will be publicly available, so you can play with it yourself. Maybe even build up your own custom data set, hopefully. But yeah, I hope that was helpful for people wanting to jump into machine learning using scikit-learn.